All right, this is a continuation of the AP Biology Podcast, Chapter 9. A couple concepts we need to cover. Different types of cells that are in our body. Some of them are called diploid cells. Diploid cells are cells that have a double set of chromosomes. That means the chromosomes are in pairs. One set of the chromosomes come from the mom, and the other set of chromosomes come from the dad. Those are called diploid. The abbreviation for a diploid cell is 2N, meaning N is the number of chromosomes. In humans, our diploid number then is 46 because we get 23 chromosomes from mom and we get 23 chromosomes from dad. They come together and that makes 46 chromosomes in each of our body cells. All body cells or somatic cells are diploid, the cells that undergo mitosis. However, not all cells are diploid. There are also cells called haploid cells. They only have a single set of chromosomes. The chromosomes are not in pairs. The abbreviation then is 1N, meaning only one set of chromosomes. Gametes are cells that are haploid for humans. Gametes are also called sex cells. Humans' haploid number then is 23. So sperm only have 23 chromosomes and eggs only have 23 chromosomes. Okay, another term we need to cover is homologous chromosomes. They are chromosomes that are similar in shape and size, and they're always in pairs. Okay, so they're paired chromosomes that are similar in shape, size, and actually they're also common, have the same information. Notice that the gene patterns are the same for homologous chromosomes. The 23 chromosomes from mom and dad pair up to become homologous chromosomes. So this is mom's chromosome and this is dad's chromosome. They pair up. Now they're considered homologous chromosomes. They also have another name in your book called homologs. This right here is showing you that where that gene pattern is on a chromosome, where the gene is located, is called the locus or loci. Another set of terms are autosomes versus sex chromosomes, different types of chromosomes. Autosomes are the 22 pairs out of the 23 chromosomes that I showed you on the karyotype from last podcast. 22 pairs, so 44 chromosomes, out of our 46 chromosomes are called autosomes. The 23rd pair is what determines the sex of the individual. Those are not considered autosomes. Autosomes do not determine the sex of the individual. Sex chromosomes do, which makes sense. Sex chromosomes are the 23rd pair in humans of chromosomes. Sex chromosomes contain X and Y, so males are XY, chromosomes do not match, and females' sex chromosomes do match, and those are considered XX. So again, here is a karyotype with 23 pairs or 46 chromosomes. All of these chromosomes are homologous. Here's a homologous pair. Here's a homologous pair. This is a diploid cell because the chromosomes are in pairs. And if you were looking at this, or the sex of this individual is a male. And these chromosomes 1 through 22 are called autosomes. So, if humans have 46 chromosomes in each of its somatic cells, how come our cell chromosome number doesn't double? How come? And that is because of meiosis. When mom's and dad's, chromosomes come together, you would think you'd have 92, but it doesn't, and it's because of meiosis. So let's go into that. So, characteristics of meiosis. Meiosis starts with a diploid cell, but ends with a haploid cell. This is called reduction division. So we're going from 2N to 1N. This guarantees a stable number of chromosomes in each generation because we're going to start with 46 and end with 23. Meiosis has two divisions, and these divisions are similar to mitosis. Meiosis creates genetic diversity. Mitosis did not. Meiosis is considered sexual reproduction because sperm and eggs are actually being made. So we have two divisions. The first meiotic division, we're going to have an interphase before meiosis called pre-meiotic interphase. So we're still going to G1, S, and G2. We're still going to grow. We're still going to synthesize and replicate our DNA and our centrioles. Then we're going to go into the first division. So the first division is going to have prophase, metaphase, anaphase, and telophase, just like in mitosis. But because there's two divisions, we have to number them. So we're going to have prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, the chromosomes are going to condense. That's the same thing that happened in mitosis. But we have something new occurring. Synapsis and crossing over are going to occur. In metaphase 1, we're going to line up. Notice they're in pairs here. In anaphase 1, we're going to separate the homologous chromosomes. The chromosome number is reduced at this point. And in telophase 1, we're going to reform the nuclear envelope, and then cytokinesis will occur. We're going to end up with 4 at the end of the second division. I guess we'll end up with 4. Between telophase 1 and prophase 2, there is interkinesis. The DNA does not duplicate again It's just like interphase. It just does not do the synthesis part. In prophase, we talked about crossing over occurs. So you got these two chromosomes here, and they're actually going to swap information. So notice, let's say the pink chromosomes from mom and the blue chromosomes from dad. At a certain point on the chromosome, they're going to synapse, which means kind of twist around each other, and it's going to break off. And the part of the pink chromosome is going to become attached to the blue, and part of this blue attached to the pink. That occurs at the chiasmata portion of the chromosome, which is just the area of where it splits off. And so what's happening is there's a swapping of genes. So the region of crossover forms an X-shaped structure. As you can see, this little X where it crosses. This X-shaped structure is called the chiasma. Crossing over creates genetic diversity because mom's chromosome doesn't have all of mom's genes anymore. It has a little portion of dad's. And dad's chromosome has some pink chromosome part to it. So that's why overall, in the end, that you look a little bit like your dad and you look a little bit more, you know, like your mom. So it creates genetic diversity and random gene mixing. That happens in the first meiotic division in prophase. In the second meiotic division, it's exactly like mitosis. Prophase 2, the chromosomes are going to condense. Metaphase 2, the chromosomes move to the equator, and the centromeres line up on the equator with the spindle fibers. Anaphase 2, the sister chromatids separate. Telophase 2, nuclear envelopes reform. And we end up with haploid 1N daughter cells, or gametes. And then those gametes are then ready for fertilization. Here is the picture for second division. Here's prophase 2, our mixed because crossing over occurred. Chromosomes here, nuclear envelopes disappearing. They line up at the spindle fibers through the centromere. The chromosomes split to chromatids and move to opposite poles by the spindle fibers. And at telophase 2, we end up with 1, 2, 3, 4. They're not called daughter cells. They're called just haploid cells. And these are actually going to develop into either sperm or egg cells. So here's our chromosome information here. We have, in meiosis 1, we have pairs of chromosomes here that are going to cross over, and then the chromatids separate into in meiosis 2. And so here's where we get our 4 new cells. Okay, if it's female versus male, we get different results. In females, this process is called oogenesis. It is the making of ovum, which is a specialized name for egg cell. In the formation of these non-fertilized eggs, only one of the daughter cells are actually going to live from a division. This division of meiosis in females is highly asymmetric. It's not even. And one of the resulting cells, the polar body, is very small. So there is a mechanism to conserve resources by concentrating it into the cell that it will undergo development into the embryo. So basically for a female, we want an egg to survive. So they put all the nutrients all into one and concentrate it into one cell, so at least they'll know that that cell will survive and then the other ones won't. Here is meiosis 2. Here's prophase 2. This actually becomes the first polar body in female. But what they're trying to show you is that out of the 4 cells that result, only one is going to survive. One egg. These 3 cells, which are called polar bodies, are not going to survive because this one is going to be larger. It's going to get more of the cytoplasm, more of the nutrients. So this one is going to survive the process. In males, it's called spermatogenesis, the making of sperm, because sperms are made. Produces 4 live sperm. Everything is even and equal. So here's a comparison side by side. In spermatogenesis, it's called the spermatogonium. It may go through mitosis to form more spermatogonium. For meiosis, we start with a primary spermatocyte. It goes to meiosis 1, and then those cells are called secondary spermatocytes. It goes through meiosis 2, and we end up with spermatids. The spermatids will go through cell differentiation and become specialized into 4 mature sperm cells with flagella. In females, oogenesis starts with an oogonium. We get a primary oocyte after mitosis. Primary oocyte goes through meiosis 1, and we get secondary oocyte and a polar body. The secondary oocyte will go through meiosis 2, and we get an ovum, an egg, that will eventually grow into a mature egg cell. Notice there's only 1 compared to 4. Going through meiosis, it provides greater variability in the genotype, which means we're getting greater variation in the environment. This sexual reproduction, this meiosis, improves ability to adapt. Diploid organisms do not merely separate their homologs during preparation of the sex cells. They actually come together. The homologous chromosomes or homologs come together. They synapse. They wrap around each other. The resulting homologs are no longer the same as the parental. Our new sperm cells are not the same as the original cell that started. They have a mixture of the genes caused by the crossing over, they produce a sex cell that is somewhat different in genetic complement from either parent. It's a mix of the mom and the dad egg and sperm. Thus, when joined with another complementary sex cell during fertilization, the zygote will have a different genetic complement from either parent. Okay? So looking at them together and comparing both, mitosis is that there is no synapsis of homologous chromosomes. There's no homologous chromosomes. These chromosomes are not in pairs. They're just individual chromosomes. Notice in meiosis, they're in pairs. See, there's a pair here and there's a pair here. Two pairs. 1, 2, 3, 4. 4 chromosomes. But they're in homologous pairs, and they're crossing over here, where in prophase and mitosis, they're not in pairs and they're not crossing over. In anaphase, these X's, these chromosomes split to half of an X. In anaphase 1, these pairs split. So this pink chromosome is going to go this way, and this blue chromosome goes this way. And they're different chromosomes than when they started with because of crossing over. This goes through regular mitosis, and in the end, our daughter cells in mitosis they're diploid. They still have 4 chromosomes, and they're identical to what was up there. And in meiosis, they are completely different because they're a mixture of the mom and the dad chromosomes from crossing over. So mitosis is most like meiosis too. Down here, different organisms time their sexual stages differently. For example, females produce eggs through oogenesis. Males produce sperm through spermatogenesis. Both of them are haploid gametes that are resulted. Fusion of the 1N gametes is fertilization. When the sperm and the egg come together, they're both haploid, they come together and they fertilize, and that always will result in a diploid 2N zygote, which will then develop into the embryo and then into the fetus, and etc. In animals, formation of the 1N cell forms a gamete, which is fused to form the zygote, which is the main form of the life form called diploplantic life cycle. So when you start with a 1N, you go through fertilization and make the 2N zygote, you are making, that's what that's called, the life cycle is called diploplantic. There are, however, many simpler organisms that do not form 1N gametes immediately. Sometimes their haploid form might be the way they are most of their life. This is true in protists like amoeba, paramecium, etc. Those guys have a life cycle called haploplantic because most of their life they are haploid, where most of our life we are diploplantic. Plants can vary. They can alternate between being haploid and diploid throughout their life, so we call that, because they go back and forth, alternation of generations. When a plant is haploid, they're called gametophyte. When the plant is diploid, they're called sporophyte. Diploplantic life cycle, we are mainly, for the most of our life, we are 2N, or diploid. For simple eukaryotes like protists that are haploplantic, most of their life they are haploid, 1N. And then plants that do alternation of generations, see, half of their life they're haploid, or gametophytes, and half of their life they are sporophytes, diploid. Timing of the formation of the zygote will vary in the lifetime, and the 2N length, how long they are diploid, varies between organisms. In plants, it's very complex.